and basic research. The interests of her lab span questions from the origin and the earliest evolution of life, using synthetic biology to colonize space, to the future of biotechnology and medicine. Dr. Kate is also the co-founder of the Synthetic Cell Therapeutics startup SynLife and coordinator of the Build a Cell synthetic cell community. Ma'am, are you here?
Hi, good morning or good afternoon.
Good afternoon, ma'am. How has your day been so far?
My day just started.
Oh,
I'm
a very
12
good afternoon
hours
then.
behind you, so
Over to you, ma'am. The next task.
thank you. Thank you. So hi everyone. Thank you very much for having me here. Thank you for the opportunity to speak to this group. I'm going to share my screen, and what I want to talk about today is a relatively new area of science, relatively weird still area of science. Basically, what we do is we study biology, but not biology as you know it. We're studying synthetic cells, which are artificial lab made organisms that we study first to understand life, understand our own biological life, and also to explore possibilities, basically to try to figure out is life can life be done in any other way than the way modern biology is doing life? And so, first, the basic question you might ask is why do we bother? Why do we bother studying synthetic life if we have the natural life to study? And there is kind of a somewhat surprising relationship that a lot of people don't think about. You look at the diversity of modern life and it's incredible. There's so many different forms of life. You're all probably familiar with this phylogeny tree that shows basically all major modern terrestrial domains of life and how they came from a last universal common ancestor. And you know that there is this incredible diversity of form, function, and biochemistry on Earth. So it's somewhat surprising that when you think about it, you realize that there actually is one life form on Earth. There is only one type of life on Earth, and that's the life that came from the last universal common ancestor of all organisms. So, when you think about it, every single biology experiment that was ever done by us as scientists from the beginning of science was done on a sample size that equals one, because we only have one modern terrestrial life. So there's one type of life on Earth. So everything we do, all of the biology experiments are done by necessity by studying that sample. So by studying modern terrestrial life. And all of you that have any science education know that sample size of one is not really the way to do science, right? So obviously, as biologists, we need to find more instances of life. And that is basically the rationale behind the whole field of synthetic cell engineering. We want to make different kinds of life so we can get out of that lineage well. We can actually study life as we don't know it. We can study life in other ways. We can study instances of life that are done in different ways than our current life. And why do we need it? Is it not enough to just study modern cells? Well, the problem with modern cells is that they're so incredibly complex. If you look at a cell, there is so many different things going on in a cell that we still don't know even the basic list of components that make even the simplest biological living cell. We can know the list of a genome to make a simplest living cell. That Craig Venter's synthetic cell, synthetic mycoplasma cell, is made with a synthetic genome. So we know the list of the genes that go into that cell, but we still don't know what every of those genes does. Even in the simplest mycoplasma cell with 400 something genes, there is about 100 genes that are essential for the living of that cell, but are not, their function is not known. So basically, a cell is a black box. We don't know what it does. We don't know how it does what it does. And we don't know all the chemical components that build it. So what we want to do as synthetic cell engineers is we want to simplify this picture. We want to build a much simpler cell. So basically what we want to do is we want to build a cell that looks a little bit like a modern cell, but is much simpler. So basically you can understand every part of that cell. And so this is essentially a synthetic cell. You can understand every single thing about that cell. You can build it, engineer it from whatever building blocks you want. And most importantly, you can make it to behave and look like this, like whatever you need to study. So this is basically a lineage agnostic biology. You can build a synthetic cell that's very modular, that contains elements of different cells, that contains elements of different clades, different kinds of organisms, and completely new synthetic elements. So you can basically make a cell that has, let's say, a membrane that comes from bacteria that expresses proteins using a mammalian system and then expresses genes using DNA, let's say from a fungi. You can mix and match elements from different kinds of organisms, from different clades. And this is the most fascinating thing about this field, is that we are not stuck to a one given lineage. We are not stuck to, let's say, studying a bacteria cell or studying a mammalian cell. We can study any type of cell we want because we build it. We build it from scratch. So here's a little bit of an anatomy of what a synthetic cell is. For those of you without kind of a much of the scientific of the biological molecular biology background, elements listed on this slide are the basic building blocks of a cell. So basically, to have a cell, you need to have a compartment and you need to have all the elements that do metabolism, express proteins inside that cell. So this is what we do. This is the kind of work that we do, which is putting cells together with all those building blocks of natural living cells. So note that I still haven't actually told you what is a synthetic cell. And the reason for that is we don't actually know because we haven't made one that's living yet. The synthetic cell, in the most kind of basic brief definition, is something that has never been alive. So this is the true lineage agnostic biology. A synthetic cell did not come from an unbroken line of living organisms ever since the last universal common ancestor of all organisms. Synthetic cell was made in the lab, and so it's an organism that's completely new. It's an organism that's not related, that's not derived from any specific living organism. And because we're all bioengineers, emphasizing the engineering part, a synthetic cell has to be controllable. So a synthetic cell is a lifelike organism that does complex functions, but we still can control any one of those functions. And this is the biggest difference between a synthetic and a natural cell. With a natural cell, we cannot control every single aspect of that cell because those cells are living and they have their own kind of plan for life. They have their own biochemistry that goes on there. With synthetic cells, we can and we do control every aspect of the biochemistry of the biology of the cell. And just to give you some examples of what we control is we control things that you normally really couldn't control in a living natural cell. And so some examples are that we can control the size of a cell over order of magnitude of size. You cannot do that with live cells. We can control exactly what information gets expressed in that cell because we make the genomes of those cells. We can control what makes the proteins. So we can control the amino acids, we can control the ribosome, the genome of the synthetic cell. And we can control the biochemistry. So we can basically control every single element of that cell. So to give you just a very brief overview of what those elements are and what do we put inside a synthetic cell, first there is a membrane. So we make a membrane from lipids that are very similar to natural lipids. So we have this lipid bilayer membrane, which is very similar to the membrane of your own cells. It's a membrane that surrounds a synthetic cell, and it's made of lipids that are identical to natural lipids. So they're lipids that are modified in the same way our own cells' phospholipids are modified. We have cholesterol in the membrane of our cells. Um, I'm sure all of you heard how cholesterol is supposed to be bad for you, and it's true, it's bad for a complex organism like us. But in a synthetic simple cell, cholesterol is actually good. Cholesterol helps to keep the membranes fluid. We can also make a membrane by assembling it in other ways than with lipids. So we can basically make it in this so-called microfluidic system, where we make a lot of synthetic cells at once in this oil water emulsion system, where we have very uniform, identical synthetic cells created in every single experiment. So this is the compartment. This is how we encapsulate the metabolism. Now, what is the metabolism of a synthetic cell? Well, metabolism of a synthetic cell is not that different from the metabolism of every other living cell. It follows the central dogma of biology. So it allows us to make proteins from DNA. And that's achieved using a cell-free protein expression system. So it basically means that we take the guts of a cell. So we take the machinery that makes proteins in natural modern cells, then we purify it. And once it's purified, we put it together with all the small molecules that we want. So all the elements of the translation system that we want to use, we put it all together, and then that's the cytoplasm of a synthetic cell. So that's how we actually make the synthetic cell perform all the actions that we want. So express proteins from genomes. And so this is what my lab is doing. My lab is basically looking at chemical processes with the context of biology. So we're looking at all the processes that a cell does in the way that a chemist would study it, not as much in a way as a biologist would study it. So we're basically taking everything that a cell does and reconstituting it with those simple components so that we can study it in details. And the reason we're doing it is because studying synthetic cells has a lot of those practical applications. If you study synthetic cells, that helps you understand natural cells. So, for example, if you study a natural biology of cancer, or if you study biology of any complex living organism, you need to understand it on a biochemical level. And that's what a synthetic cell lets you do. If you reconstitute a certain pathway from, let's say, a human cell in a synthetic cell, that helps you study it with a lot of detail. That helps you do a very kind of careful consideration of every part of that pathway. You can also build tools for synthetic cells, and by building those tools, you're able to understand natural cells much better. So there's this feedback loop between making tools for synthetic biology and using those tools to study natural biology. And then there's a lot of applications of synthetic cells. And I'm going to show you examples of few of those applications. Using synthetic cells, you can study biocomputing. So you can basically build synthetic cells that act as little biocomputers, little biochemical computers that can either interface with natural cells or interface with electronics. You can study the origin of life, origins on Earth, and origins of life elsewhere in the universe. You can build tools that help us plan space exploration. You can use synthetic cells as little biomanufacturing machines, little biofactories that let us make drugs on demand in space. You can study metabolic engineering, which is a basically a process of assembling enzymes that make whatever transformations you want. So this is the way to make drugs. This is the way to make new materials. And you can also use synthetic cells to study biomedical applications. They can be used as drugs. They can be used as diagnostics and therapy tools. And I'm going to show you a few examples of what my lab is doing to get there, what kind of things we're doing with synthetic cells. So one of the kind of biggest areas that we study is how cells assemble and how cells can mate and differentiate. So this is this basic process that we know from living cells that was really hard to reconstitute in natural cells. How do you work on complex genetic circuits that build genetic pathways of increased complexity? And this is one of the things we do is make synthetic cells that can mate and then make synthetic cells that can differentiate. So mating is this process where two genomes connect, and that's what our synthetic cells do. And then differentiation is this process where one lineage of a synthetic cell becomes two or more different lineages. And what you see on this graph is a schematic of how a synthetic cell populations can mate and differentiate into new lineages. And one of the applications for it is we can control genetic circuits. So genetic circuits that are very similar to the Boolean logic gates that you know from computer science. And the application, one of the applications of that project is obviously to use it for biocomputing. So that's kind of down the line. That's something that we want to do eventually, is to be able to use synthetic cells to engineer those kind of little biocomputers, little processing machines that work on biological parts but give you the same logic gate operation freedom as electronics does right now. And another application of this technology is you can have a synthetic cell that differentiates into different lineages. And so what we do is we make synthetic cell that, as a result of complex genetic processes inside it, differentiates into different lineages. And those lineages can either express different kinds of proteins, or they can produce different small molecules. So in that way, the synthetic cells are pluripotent, which means they can turn into any kind of cell, any kind of lineage that you want. And the way we do it is by this stepwise fusion or mating of synthetic cells. And the way it's done is we mimic the natural processes for fusing of compartments. So what you see on this slide is a DNA representation of what a biological fusion of two lipid membranes looks like. So in nature, nature does it with proteins. We have proteins on surfaces of cells, and those proteins bring together membranes, and those membranes fuse. We mimic those processes with DNA. So we fuse cells as a result of fusion of those DNA oligos, DNA dimers. And what comes out of it, I'm not going to go into experimental details for the sake of time here, but at the end, basically what comes out of it is the ability to differentiate cells into different kinds of lineages. And so what you can do with it is you can have those sequences of synthetic cells that you can programmably and in a combinatorial way differentiate into different kinds of lineages, different kinds of cells. And why do we want to do that? Well, for one, we can program genetic pathways in synthetic cells. So we can have synthetic cells that start to approach the complexity of natural cells. We can make synthetic cells that grow, differentiate, that kind of make more and more different complex pathways. We can have synthetic cells that mimic the cells that existed on early Earth. So we basically use this process to study the origin of life on Earth, how life started on an early Earth. And we can study some fundamental concepts that let us one day hopefully build something like a biocomputer. For example, we can study how information flows in cells, how synthetic cells can mimic those complex natural evolutionary processes. And another area where synthetic cells and their applications are really useful is studying things that cannot be studied in live cells. And one of those things that are very hard to study in natural cells is a ribosome. So ribosome is this essential, central to all life enzyme, this enzyme that makes proteins. You might think ribosome is a very efficient enzyme because every organism on Earth uses it, but that's not true. Ribosome is actually a terrible enzyme. All of our proteins were made by this crappy enzyme that's very slow and relatively low fidelity, except to all those proteins that later ensure that the new protein chain has the right fidelity. So basically, the ribosome itself is a molecular fossil. It's a very old enzyme. It's as old as life on Earth. The catalytic center of the ribosome, so that part that makes proteins in a ribosome, was established and did not change since life started. So looking at the ribosome, you're basically looking at that molecular fossil. You're looking at exactly how life looked like at that very time when life started. And that's an advantage of synthetic cell technologies, is that you can study something that's so old and so fragile. And the way we do it is we study a ribosome by breaking it. That's how, if you want to figure out how something works, you want to break it apart. And so that's what we do. We break apart the ribosome. And the reason we can do it in synthetic cells is because they have much wider range of conditions under which they maintain fitness. So basically, in a live cell, if you break a ribosome, you end up with a dead cell because the cell needs the ribosome, and ribosome is already a bad enzyme, so there's no slack. If you break a ribosome, the cell is dead and you have nothing to study. In a synthetic cell, you can break a healthy ribosome and the cell will still limp along. You can still have the cell move on. You can have the cell make proteins in arguably in small quantities and with low fidelity, but it's going to work. So you can basically make things in a synthetic cell in a way that you wouldn't in a natural cell. You can break ribosomes and you can make it into a kind of new kind of a protein synthesis machinery. And so we're doing it to figure out how the oldest ribosome could have looked like. So we're making a simplest ribosome, but we're also trying to evolve it forward. We're trying to make a better ribosome. We're trying to make a ribosome that helps us figure out how, what could be the future of biomanufacturing? How could we make a ribosome that lets us make more complex molecules while still using this biological machinery? Another thing that synthetic cells can be used for is making things that you then export outside of them. So you can imagine a synthetic cell making a protein or a drug that then gets exported outside of that synthetic cell. And if you can export something outside of a cell, then the next thing is, can you envelope it into another cell? And so basically you're looking at the origin of multi compartments inside the cell. So basically origin of a eukaryotic cell. So when you think about it, when you look at the evolutionary history of life, there were those boring billions. We call them boring billions because nothing really happened for about 2 billion years on Earth. Life started, and then that was it. Life started and just stayed as a unicellular, simple organism for literally 2 billion of years. And then something happened, some evolutionary transition, evolutionary phenomena happened, and life just exploded in variety. And so we want to study that transition. We want to see how synthetic cells can come from those simple unicellular or prokaryotic-like, so bacteria-like cells. How can they become more complex? How can we make organelles? Basically, so what's the origin of organelles? And how can we study how complex cells came to be, because all of our cells, we're obviously eukaryotes, so we have all this incredible complexity inside a cell. Using synthetic cells, we can study that transition. We can study how organelles came to be and what are the easiest ways to study this kind of molecular basis of organelles. And then another thing that's possible to study with synthetic cells is how synthetic cells communicate with natural cells. This is useful for some practical applications that I'm going to show you in a moment. For example, this is the basis of our International Space Station experiment, where we're going to use synthetic cells to control natural cells. But this is also the basics for studying how cell collaboration came to be, how cells came to live next to each other, how cells came to kind of work with each other, how populations of cells emerged to work together. And here's an example of how it looks like. If you co-culture synthetic cell with a natural cell, in this particular microscopy image, the red is synthetic cells and green are natural cells. We can co-culture those cells and study the interactions between those cells. So we can study interactions between natural and artificial cells. And one of the applications of that system is that we want to be able to make biomanufacturing on demand. So we can basically make compounds in conditions under which normal complex bioreactors could not function. So we make complex chemicals, or we make biologicals that can be drugs, that can be nutrients, by using synthetic cells to control natural cell bioreactors. And one of the kind of obvious applications of it is in space. When you travel into space, you want to be able to make compounds on demand in a short amount of time, on short notice. And so this is one of the things that synthetic cells are really good for. Another thing is, so I talked about this, how synthetic cells can communicate with natural cells, and how can we make organelles in synthetic cells. And one way to combine it is to make organelles that are made of natural cells inside synthetic cells. And so what I'm showing here is this kind of a funny idea of using a natural cell, in this case, a cyanobacteria cell, inside a synthetic cell as an organelle. So a cyanobacteria is trapped inside the synthetic cell, and that cyanobacteria acts as an organelle. What does the organelle do? Well, you might ask. Where in this case, what does organelle does is it makes a synthetic cell grow. So what you can see on this video is you have a synthetic cell that grows as a result of lipids that are produced by a cyanobacteria that's encapsulated inside that synthetic cell. So another thing that we study in synthetic cells is we want to see if we can recreate simplest natural cells. So right now, the simplest living cell is a organism called Mycoplasma laboratorium, which is basically an engineered mycoplasma cell that Craig Venter Institute engineered by minimizing natural mycoplasma. What we want to do is we want to take the genome of that Mycoplasma laboratorium and put it inside a synthetic cell. And once you do that, will it actually start being alive? Will it actually start replicating and synthesizing all the proteins of a natural cell? This is work in progress, but if that works, we would be able to say we actually took something that's not living, a simple synthetic cell, and made it living. So we basically put all the elements together to create a living cell. So I've been talking about how synthetic cells are really simple. So a nice thing about simple cells and simple biological experiments, or simple experiments of any kind, is that you can digitize the information about those cells. So for example, you can take information about a synthetic cell, digitize it, so translate it into this information needed to make it, and send it to a remote location. So you can make a synthetic cell in one place in the world, and then by digitizing the information and sending it to a remote location, you can recreate that synthetic cell experiment in any way you want. So basically, as long as you agree on a library of parts, one lab can make a synthetic cell and then wirelessly send it to another lab, information about making that cell, and that other lab can recreate it. You cannot do that with natural biology. If I make a bacteria strain, I have to send you that strain so you can have it. If I make a synthetic cell, I can send you information about making it, and you can recreate that synthetic cell within 6 to 12 hours. This has a lot of applications for remote medicine, for point of care medicine, where you want to make certain therapeutic system on demand. But when you think about remote locations, it doesn't get much more remote than space exploration. We're using this technology to develop solutions for long-term space exploration. So for making drugs on a spaceship during long-term space missions, where we have a support lab on Earth that creates something, that creates a drug or a synthetic cell that makes a nutrient, and then we send information about making that synthetic cell to a ship or a colony that's equipped with a DNA synthesizer and all those biological parts to make a synthetic cell. And then they make a synthetic cell that then acts in a certain way. And one of the kind of obvious applications of it is that it creates this tie between space biology and astropharmacy and medicine. We can use synthetic cells not just to study life, but to help us explore solar system, to help us build kind of long-term human habitats before they become self-sustaining. And then quickly going back to basic research, if you're looking at a this incredible diversity of life, there is a lot of different kinds of life, but we don't actually have a one kind of common integrated way in which we can measure if something is alive. So there's this one project that we're working on right now is how do you measure life? How do you quantify life? How do you quantify aliveness of a system? This might sound like a simple academic question that doesn't have any kind of practical applications, but it surprisingly actually does have practical application. We are already sending probes to other bodies in a solar system. You know that just a few weeks ago, we had this touch and go of an asteroid where we collected samples from a surface of an asteroid. We have sample return missions planned for Mars. We have missions planned for the next few decades where we're going to go to other places in the solar system where life is likely, where we're going to go to Enceladus, we're going to go to Europa. These are all the places in the solar system when life could possibly happen. And so if you go there, let's say you go to Mars and collect a sample, how do you know that that organic matter that you get into in your lab has ever been alive? Because it's going to be an organic matter, but organic matter can be synthesized abiotically. The question is, how do we make this kind of life meter? How do we measure aliveness of a system? How do we measure if basically a tube of goo, if something that's a biological matter was ever alive, if it's coming from a living system? We don't have a good way of doing it right now. And so that's where the synthetic cell project comes in. We use synthetic cells to build organisms of increased complexity so we can study this transition between simple biochemical experiment and a more complex living cell. And one of the fields where it's really important and when it's really become applicable is not just looking for life elsewhere in the solar system, but also looking for life on Earth. It sounds counterintuitive because you're going to tell me, of course, there's life on Earth. And I, as a member of that life, agree with you. Yes, there's life on Earth. But there are environments on Earth that have been separated, have been physically separated from other parts of our biosphere for a few billion years, really. Things like deep frozen lakes, very deeply into the ground, very deeply under some ice in certain parts of Earth, in some parts of desert on Earth. There are environments where it's possible that an alternative origin of life gave rise to this so-called shadow biosphere. So it's possible that there is a biosphere on Earth that's not us, And in order to find it, we need to know what we're looking for. And that's another application of building more and more complex synthetic cells, is that we can go and look for that biosphere. So this was kind of a very, very brief overview of all the applications of synthetic cells for fundamental science. I want to leave you with this kind of interesting and upsetting graph. This graph shows the amount of funding that we put into biological R&D versus amount of drugs that we make. And you can see it's an inversely proportional relationship. The more money we put into developing new drugs, the less drugs we get. And so there's clearly something wrong with it. There's something fundamentally wrong with the way we do biological research and drug discovery right now, because the more resources we put in, the least amount of drugs we get out of it. And so one of the ways to solve it would be to look for a new chassis, look for a new way to making drugs, the new way of studying biology. And we propose that synthetic cells could be that way. We could use synthetic cells as this new chassis to study biology in order to learn more about either modern biology or biology as we don't know it. And to put it in context, synthetic biomedical applications of synthetic cells are already pretty well developed. For example, synthetic cells can be used to shrink tumors in live mice. And now I'm going to show you an example of this astropharmacy, so basically making medicine for space exploration that I was discussing before. So the idea is that you want to make a set of components, launch it into space, and then make drugs when they're needed in small quantities that are ready to administer to a patient. And so as a result of this fantastic collaboration with people that build space architecture and people that make drugs, we built this mission where we sent 2 different synthetic cell payloads into space. And so what we did is we built this little QB-shaped self-contained experimental chamber that contained 2 different experiments, an RNA folding experiment and a protein expression experiment. So those 2 different experiments help us understand what exactly is going on when you actually launch a synthetic cell into space. And so what we did is we packed it up, we put it on a rocket, and the rocket went up. This is one of my favorite videos of all time, because this is actually the first time I saw my own payload going up into space, which was really exciting for me personally. And I also had to get up really early for it. So you get up, you see your rocket launch, and then you hope it doesn't crash, it doesn't burn. Ours did not crash, did not burn, so I'm obviously really happy about it. We managed to send up a payload, and that payload then came back. It was a sample return mission. We managed to analyze that payload in the lab. And now what we're doing is we're sending a more complex payload. We're designing an experiment that can actually be performed by an astronaut on the International Space Station to make sure that we can have those kind of complex synthetic cell machinery made. And so this is kind of the summary of the research part of my talk. Synthetic cells are awesome, people. You can do a lot of things. You can learn about biology. You can make new things. You can build new tools. And so hopefully in the future, we'll be able to actually make really complex, really interesting things out of those synthetic cells. And the way we want to do it is we want to do it as an international community. And so here's kind of a provocative thought experiment. We know that some international research communities can come together and command huge resources for their work. We can study galaxies using those billion-dollar worth telescopes. We can study particles by building those large particle colliders, high energy particle colliders that also cost billions. So it's possible for research communities to come together and from governments and from private agencies, for private funders, command those huge resources for the entire world scientific community. So the question is, what would be a synthetic biology, synthetic life equivalent of such huge resources? I don't have an answer to that. That's more of a thought experiment. What would we do if we were able to come together as an international community and build artificial cells together, study artificial cells together, and make all those applications that I was discussing actually come true? And so we're working on it as an international community. I'm a co-founder of Build a Cell, which is an international synthetic cell community. We're a group of people from all over the world that study engineering life, study engineering cells from scratch. And I just want to note that we actually don't have a Build a Cell member lab from India yet. So if any of you know a lab that works on synthetic biology or on engineering cells, Build a Cell is an open community. You can join us. You can join anytime. You can join without any consequences. So you can join and do nothing, or you can join and become a really active member of the community. You can also join even if your lab is not a member. We have right now hundreds of individuals that are members. Membership is absolutely free and absolutely voluntary. People can come to our workshops. People can basically interact with the community in any way we want. And the goal of it is to build this unified community that studies cells. What we do is we meet often, we meet online, we meet in person, we produce protocols, we're coordinating research efforts, we're coordinating outreach to the public, we're coordinating science policy, we're coordinating outreach to government agencies on biosecurity, on biosafety of synthetic cell research. We also have those meetings twice a year. The whole Build a Cell community meets. Next meeting will be in February of 2021, probably again online, remotely, which might actually be a good news for some of you who want to join, because you can join remotely. Meetings are always free. You can always join if you want. You can always drop out anytime you want. It's an absolutely voluntary community of people that want to build cells. And so my time's running up, so I want to say thank you to all of you. I want to say thank you to all of my lab. All the research that I've shown would not be possible without the fantastic people that work in my lab and without all the funding agencies that support our work. So thank you very much. And if there are any questions, I would be happy to take them. I will stop sharing.
All right. The chat actually.
Oh,
would you like me to read them for you?
Oh yeah, I actually see them. I came out of chat, or you can read them for the recording.
All right, I'll just read them out. Are you doing some research on the photosynthetic therapy in your lab?
We are doing research on assembling a photosynthetic system. So we're basically doing research on building a synthetic cell that can do photosynthesis. We don't do any therapeutic research in our lab at this point.
Geoffrey asks, wow, great work going on, very interesting and promising. How do you guard against any biosecurity issues and dual use of this synthetic cells?
This is a very important question and a very difficult question. Thank you very much for it. Right now, synthetic cells, because it's such a new field, we don't have a even national and not to mention international biosafety, biosecurity policy. We're working on it. That's one of the things that synthetic cell community of Build a Cell is working on. We're using guidelines from other areas of synthetic biology to build some sort of a BSL2 classification and kind of good usage policies. It's really hard to guide against dual use. This is something that all of synthetic biology community is dealing with. It's really hard to guard ourselves against bad actors without compromising the integrity of the research community. So basically, if you do research, it has to become open source. It has to be available to everyone, because otherwise there's no progress. But obviously, the downside of it is that if we guide guard ourselves too much, then we won't be able to admit new members, we won't be able to train students. But if we're too open, all this information is out there in the open for bad actors. And that's, unfortunately, there's no good answer to that. All of synthetic biology is struggling with it right now, is how do we keep research open but safe? Within synthetic cell community, we're working on it. We're working on those guidelines of what kind of research should be supported and what kind of research probably might be better not be done. And we're trying to keep it safe, obviously, within our own labs. We're trying to make sure that there are biosafety, biosecurity considerations within our own lab.
That was something truly to think about. Next
you.
up, we have Mr. Mehfoos. He asks, can we make synthetic cells-based organism? It may be a taboo, just asking out of curiosity.
It's absolutely not a taboo. I think anything that's not actually wrong should not be a taboo in research. And so right now, the technology is not yet good enough to make a multicellular organism, but we're working on it. We are working on origin of multicellularity. I didn't show any data from that project because it's still very early stages, but we're working on being able to have synthetic cells connect with each other, talk to each other, and that's a first step for multicellularity. And other groups are working on it as well, developing kind of synthetic tissues, building tissues out of completely engineered cells. And so one day, hopefully, we'll be able to say we can actually make a multicellular synthetic organism.
Okay. I hope Mehfoos got his answer. Next up, we have Kashish, who asks, what is the survival rate and viability of synthetic cells? Aren't there any compatibility issues among the various components taken from organisms of different domains of life? Like we see in plasmid incompatibility within different bacterial strains.
Thank you. That's a great. That's a great question. So the survival rate is pretty high. Viability is depending on how you design them. They can be viable for anywhere between 12 hours to several days. Right now, the limit is about a week. After a week, we're running out of components that lose stability over time. There is very little actually issue with compatibility among various components taken from different organisms because we designed this system from scratch. That's one of the advantages of using synthetic cells over natural cells.
Okay, next up. We have Raymond asks, just to be more clear, how is synthetic biology different from genetic engineering?
Okay. How synthetic biology differs from genetic engineering? It doesn't really differ. It's kind of part of the same research. It's part of the same kind of research enterprise. They're different tools, different techniques, but they're not different.
Okay. Next up, we have Sudipta Das. Ma'am, if you don't mind and don't have any problem, can we get your PPT file? It will be a great honor and achievement for us to get such kind of presentation.
Thank you. Shoot me an email and I'm happy to share.
Okay, so then there is Anushka More. Synthetic cells can be programmed and implemented on the pancreas to treat diabetes?
Right now, no, we don't have that technology, but it is one of the things that people in our field are working on, using synthetic cells to recreate the cells that produce insulin. It's not in clinical trials yet, but it is one of the projects that I know are definitely going on.
Okay, then we have Aman Bhargav asking, how challenging is it to maintain the components without a cell wall?
So not very, actually. Cell wall increases stability and rigidity of cells, but our lipid membranes are stable enough. You can have those synthetic cells go on for days and they're pretty stable. it's all about the membrane component. It's all about the composition of your membrane. So you basically need to pick the membrane compositions for the environment that you're working with. But once you do, most of the time it works fine.
Then we have Dr. Silpi asking, what is the criteria to join the lab for young researchers who want to learn the skill of understanding the concept of synthetic cells? Can anyone interested join, or is there any specificity?
Anyone can join. I always invite everyone to join this field and to join my lab. I recruit graduate students from 2 different programs. If you go to my website, those programs are listed there. So anyone who wants to work on it as a grad student can apply to those PhD programs. We have undergrads from the University of Minnesota, and we are also always looking for postdocs. So anyone on every stage of the career is always welcomed.
Okay, next up we have Adrian. He asks, is it expensive to engineer this type of synthetic cell?
Surprisingly not. Expensive obviously is a relative term. Engineering synthetic cells is actually cheaper than engineering natural live cells. It's not, cheap enough to do in your own home, but it is less expensive than biological research on, let's say, mammalian cells.
We have Khalid asking, have you found any certain chemokines which these cells use for interreaction?
We engineer those chemokines. So because the whole system is engineered, we don't have to, discover the chemokines because we engineer them. So we have a lot of different kinds of small molecules that they're mostly small membrane permeable molecules and short DNA oligos that different populations of synthetic cells use to interact with each other.
Okay, so then we have Winil asking if there are any remote volunteering opportunities available for your organization.
Anyone can join Build a Cell. So if you basically Google Build a Cell and go to our website, there is a working group page where you can see all kinds of working groups that we have, and everyone is free to join any of those working groups. So if you have interest in any of the activities that Build a Cell does, even if your lab is not a member of Build a Cell, even if you're just undergrad and don't have a lab, you can always join. So you can go and basically email the leader of any of those working groups listed on a Build a Cell website, and they will accommodate you. They will find something for that kind of fits your expertise and fits your interests.
All right, then I think we're out of questions now. Audience, if you have any more questions, I'm sure she'd love to hear from you.
Yeah. So thank you very much, and thank you for having me.
Thank you so much for coming in today. It was an honor having you.
Thank you.